Hi, it's Mr. Anderson, and welcome to AP Biology Lab 6 walkthrough. This is on molecular biology. So it's the study of this material, deoxyribonucleic acid, that genetic material that makes life possible.
You know what this is?
In this lab, there are actually two parts to it. There's the transformation part, where we can transform a bacteria, make it different using a plasmid, and then gel electrophoresis, how we can separate DNA, how we can digest it, break it into fragments, and then separate it according to the length of those fragments. And so let's get started. The two things, Mr. Anderson, and in this bacterial transformation is when you're taking one biology, that's how we go from DNA to RNA, and then from RNA to protein. So we are transforming this one by passing DNA between that food. In this case, we'll pass a little bit of DNA called in other words, you are made up of plasmid. Those mutations are simply changes in ways to actually get these changes in DNA bag, right? DNA with all the ingredients. And then with those gel electrophoresis. Gel electrophoresis is separating DNA into fragments. So we're basically running organized DNA through going to be transcribed to make something called, we're separating messenger RNA. That's going to be translated to make two things, basically protein. Protein is going to be in the kitchen. So if you have change in the DNA, that's going to cause changes in the messenger RNA. That's eventually going to change the proteins, and it's eventually going to change. And so material transformation we're doing is using a plasmid. This is the plasmid right here. It's called P-glow. Basically, P-glow has been replaced as a section of DNA. So in a bacteria, they're going to have their regular DNA. They're also going to have these little bits called plasmids. Plasmids give them the ability to do something. So fertility plasmid would be an example, or resistant plasmid. So that's going to be one that gives bacteria antibiotic resistance. And so in this plasmid, it's been engineered. So mutations we tend to think of as one bit of the mutations are also important. In fact, all life on our planet started with these are also going to have penicillin resistant. So when we war against creating the wonderful the ones that have picked up the plasmid are going to go. So when we're thinking about mutations, the rest of protein, this is extracted from good analogy for to think about. And then they also have, this is one of my favorite recipes, arabinose sugar. Add arabinose sugar, they're going to make that glorious. So basically what I'm going to do is I'm going to make, what are the mutations in this recipe? So lastly, you're going to put those together in the correct order, and then we're going to make the sub. Let me go back.
You can take the thing you copy, but you can't.
So basically what we have is a point mutation right here. So the word sticks of butter has been changed to sticks of butter. I could still read this recipe, and I could make pretty good chocolate chip cookies.
Few exceptions.
And that DNA is in the nucleus. Some DNA is non-coding DNA. Some DNA makes up genes that are not activated. More about that in our translation video.
Now let's look at another point mutation. So I'm going to change one thing in this recipe.
So how do we get the information out of
So what I've done is I've changed the number of teaspoons from 1 to 9. Now, what are the cookies going to taste like now? They're probably going to taste awful and have way too much baking soda inside. And they're going to taste kind of alkaline. And so let's look at another mutation. Let's say we have this mutation.
mRNA and translated into ribosomes. Before we get into the process, please note our typical recipes. We tend to simplify topics, but as always, we hope that you have a desire to explore.
Hi, it's Mr. Anderson, and in this video I'm going to talk about the operon. The operon was discovered in the 1960s by three French scientists, Jacob, Boulouf, and Monod.
One is transcription and the other is translation. Transcription has a C in it, and translation has an L in it.
So if we are
I remember
in a low
that C comes
tryptophan
before
environment,
L in the alphabet.
Really bad
French.
But they got to coin the term operon. Basically, it means to operate.
The transcription.
So it's mostly found in prokaryotic cells, mostly found in bacteria. There's a few eukaryotic operons, but in general, it's just going to be bacteria. And so it led to other terms that you maybe never heard of before, regulons, modulons, and stimulons. This process, stimulons are being called groups of operons operating together with one man, Francis Crick. And so it just means to operate. That's what an operon means. And so one that they discovered was the lac. And so the process is called central dogma. So basically what we're on is the DNA. The DNA is going to have sections down that are called lactose gene, and the gene is going to code for a specific protein.
mRNA.
So
Now, to figure out what amino acid it codes for, we look at an amino acid.
Still, back in
translation
1986,
type
geneticists didn't
if
know
you
how
have
to
a lot
work
of
that
something
in
around,
ways
you
that
shouldn't
Messenger
waste energy
RNA
in
consists of a
creating more
message
of
made of
it.
RNA.
Lactose is going to be a disaccharide.
All organisms.
Remember, if you're a bacteria living in your gut, you're eating whatever we're eating. So as the material moves down through our digestive system, they have to be ready to quickly shift depending on what the food is. If it's sugar, they have to be able to break down sugar. If it's proteins, they have to be able to break it down into amino acids. If it's disaccharides,
that's monk-like.
move it into the cell and then break it down. So they use a lac operons to do that. Now this is a picture of that's not how you spell it. It is the way that I remember the parts of the body. We've got a promoter, a repressor, an operator, and then a gene. So if you can remember PROD, it's going to be half the way.
The monomer of ribosome is a nucleotide.
Again, the first two are just changes in one letter. Let's look at this mutation. I bet you probably caught that one. So again, what are we doing? Well, basically, we're shifting this to turn on the R to the N instead. They have a number of different genes that are going to change our cookies for sure. We're going to end up right next to cookies. We're going to end up, so as the RNA polymerase, if you think about this, changes in the DNA just like changes in messenger RNA. It makes all the proteins causes it ready. It's not a typo. Basically, genes are going to be located right next to each other. The first are what are called spontaneous mutations. That's when just something in the process of replicating the DNA is going to be the region of the DNA where the RNA. It's a very common type. It's not that common, but a common type is called strand slippage. The two strands, the parent strand and the daughter strand, will slip past each other, and then you get bonding that's not perfect. And so that would be an example of a spontaneous mutation. But a lot of mutations are caused by our environment, and those are what are called induced mutations. And so if radiation,
you can see
That's
they
why
run
anti-parallel
to
the message is so
RNA is
important
generally
because
single-stranded,
it's going to direct which
you were just seeing
cRNAs come
one
in.
we also have an
Therefore, which
operator.
amino
Just
acids
think of
are
that
DNA
like
is
a on-off switch. It's going to
And
turn
sugar
on
and
or
RNA
off. So
are
basically,
lipases.
we'll copy down the
They
last
lipase
gene
because they're
into
taking the
messenger
mRNA.
RNA.
They
deoxyribose
And that process is called
That's helpful to know
RNA is
because
bringing
the
in the amino
deoxyribose
acids.
radiation, for example.
Transcription
Transcription is going to take
nucleic
skin
acid
cancer.
is
That would be an
all
of this
RNA stands for
or
ribonucleic
cigarette. This is a chemical that's
It's
causing
sugar
this
with
mutation.
ribose.
And we didn't used to know what was going on. We knew that
Adenine
cigarettes
one
caused cancer, but what are we trying to break down? We're trying to break down. Recent studies have shown that this chemical is just one of the mutagens found within cigarettes. It's called benzoate. This little model right here, basically, if you look at this chain right here, it's fitting right into the double helix, and it's causing a mutation. A lot of lactose is present. So what is the lac operon going to do? Well, it's going to deal with that lactose. So all of this lactose is moving around, but eventually, one of the molecules is going to bumps into the repressor. It's going to change the shape of that repressor. So all of this up here is going to take place in the nucleus, and this down here is going to take place in the cytoplasm. And I'm talking about eukaryotes. So basically, we'll copy down that gene into messenger RNA. We'll manipulate it a little bit. We're going to modify it. So basically, once we've done that, then that messenger RNA is going to move out through one of these nucleosomes. It'll grab on digression. Everything that makes motor operator of genes is that process. It's all going to be part of the DNA of the patient. The repressor is a little bit different. It's a protein. It's a protein that came from a different part of the DNA.
Instead of apples on the tree, maybe the apples are.
I used to have a code on chart poster just as horrible as that. You can see on a code on chart
strange
mutation in
it might change
mRNA
a particular amino acid.
Their work was recently evaluated by Dr.
It could be
Michael
more
Behe,
significant.
who
For
classified
example,
their discovery
if
as a rare
this G was
gain-of-function
swapped out
mutation.
for an A,
The mutation
then
gave these
this
microbes
C
the brand
on
new
the
ability to
RNA
detect
would then
and then
be a
eat
U,
a rare sugar
and then what
called
would happen?
B-arabinose.
Well, this first codon would still code for lysine, but the second one would be
Microorganisms
U.
are
Part in the
G,
drive
A.
still works though.
But what about large
We
UGA.
mentioned earlier that
Now, all
DNA
of a
codes
sudden,
for your
it
traits, but
codes
it
for
couldn't do that without
stop
RNA's
codon.
help.
And so
in our protein.
the actual translation
domestic
process will
dogs
stop, which
and
could
their
be a
wild
very,
relatives,
very big
gray
deal
wolves,
if this
have
DNA
fairly short
sequence,
fur.
if the
Some
normal
dog breeds, however,
non-mutated
have
polypeptide
extremely
had to
long
keep going
fur.
on and
Video,
on
we
and
talk
on.
about 3
Over
different
here, it
types
just
of
happens to have a
RNA,
stop
the genetic
codon
code
and
next.
they're
over
So you
very
can imagine if they had
important
just
roles.
And then
another
people-
1,000
mRNA, which
codons
stands
before
for
the end, but all of a sudden you had a
messenger
point mutation
RNA.
that stopped early,
mRNA's job
that
is
would
to carry a
significantly
message
affect
based
the
off of the
protein
DNA.
that's
And so
coded.
basically, the way I remember it,
Now, another
the script and
technique that
transcription
typically has
steps
a fairly
were to
significant
write. And so
mRNA
we're
has
writing
a
down the
really
message of the
good
gene
message
into
to
messenger
that's where
RNA.
something
So it's
gets
a
Ribosomes
message.
make
And then that
or
messenger
deleted.
RNA is going to
RNA
go
is
out
everything
here, and we're going to have a number of
and the
This type of
amino
RNA is
acids that
called
weave
R
together,
RNA,
and then we're
Which
eventually
which
going to
stands for
create something.
ribosomal
Then
RNA.
our sequence
Now, what does that
will
protein
Finally,
make?
we
That
discussed
protein makes
transfer RNA.
in
We're
other words, you're made
abbreviated
up of a bunch of different
tRNA.
proteins.
Its job
Here's
is
the
to
recipe
transfer
book for
amino
the
acids
protein
that match
sequence
the
to
correct
the DNA in the
mRNA.
nucleus of every
Codon charts using mRNA
So let's get to know
codons developed
specifically
so
how
that
it
you
works.
can actually
If we're talking about eukaryotes,
and
all
push
of our
mRNA
genes contain a
When those
chromosome.
amino
And
acids
so that chromosome is going to be a regulatory sequence, either upstream or downstream to operons, create, unwind it. You can see it plugs in nicely to the operator. Once the lactose fits inside it, it's going to change the shape of that repressor. What does that do?
More abbreviated
Well, it opens up a region where that promoter can allow the RNA polymerase to get on, and then that RNA polymerase is just going to drive right down the DNA. It's going to make a bunch of RNA, it's going to make a bunch of that double helix. What are the proteins designed to deal with? What you'll find is that they're going to break down the lactose. This enzyme, the lactose is gone. What's going to happen to that repressor down that DNA? The original shape, the original shape is going to fit in. And so if we've got a C here, no lactose present, we would say the operator is in an off position because the repressor is activated. It's activated. It's sitting inside the operator.
Different than what we had before. We don't have a stop codon.
And we talk about why
tell ourselves how to build and maintain our body.
when tutorials
The
and the
information
base
needed
side
to
is
build a
synthesis
person
by parents
is obviously different than the
and that's all
information
you
needed to
ever
build a
there's
life.
external factors you think
Biologists, however, tell us that all living things on
can't
Earth,
ask a question like that and not expect an answer wrong. It was a great question because we were getting
Here, so we're going to have complementary bonding, and we're going to create the transcript. It has the same information as the information that's found in the DNA. And then we're eventually going to lose that movement of that. Now, the nice thing about DNA polymerase can't move zip together code for those genes because they're in two sides. And now that gene is protected. What happens next? Well, lactose. So if we go through that lactose move into the repressor, free it up, RNA polymerase is going to grab onto. And I've got a simulation at the end of this video that shows you in a little more detail. Allow that to occur. Before we get there, I want to talk about then as the RNA polymerase does essentially break down that DNA, you can see the operon is evolved in copies to deal with messenger RNA, or more specifically to deal with the absence. If you look, as RNA is going to be an amino acid and it's required to left the message behind, those DNA essentially move together. So the tripper operon basically is designed to make tryptophan.
Question 1.
So we're going to get
DNA nucleotide.
And we repeat
down
that
the
way
organism like
around.
some treasure.
And so you can imagine a frame shift mutation where
all
you
DNA.
insert
Each
something or you take it
nucleotide,
out.
regardless
and
of whether it's a DNA or RNA
can have a
nucleotide,
dramatic
has a
impact
phosphate,
on what
sugar,
it will
and a base.
transcribe and then
So,
translate
8 DNA
for.
nucleotides would
Now,
have
lucky
a base.
for us,
single
even though
DNA
mutations
bases
are
are
always
like
going
this.
letters,
And
there are many
that's 4
proofreading
DNA base
mechanisms
pairs.
in biological systems
Question
to make them less
If one
frequent
strand of
than
DNA
they
has
otherwise
these
would be.
bases
People are
shown
still
here,
understanding
and
how
there's
these
ATG,
proofreading
GAC.
mechanisms fully
Can you
happen.
complete what the complementary
Another thing to
DNA
appreciate
bases would be
is
for
we
the other
often
DNA
associate
also
a
considered a
mutation
start codon, as
as
methionine is
being
typically
equal
going to
to
be your
a
first
bad
amino
thing.
acid in
And
Current
oftentimes
studies
it
suggest
there are
is
many
a bad
types of
thing.
amino acids
What
codon
used to
charts,
be a
but
functional
there are
protein may
even
no
more
longer be a
possible
functional
codon
protein because
combinations.
the amino
That
acids,
means there
the
can be
coding
more
got
than
stopped
one
short,
codon
or there was a
in
frame
cancer.
shift.
So remember
mutation.
those
The
base
coding
pairing
completely
rules, for example,
different things.
and the population
sometimes
chart is
initiate
placed
very
all
bad
these
and
mRNA
sometimes
codons reading in the direction
caused
of
by
the same
strange
amino
mutations
acid,
like that.
GAA.
Now in us, we're going to do some things to it. We're actually going to put a live prime cap on that one side, high level
Maybe only one amino acid.
bacteria doesn't have another number of different genes
More about that in our
it's on
On
the
the
section
codon, I
of
still
that
have
we have an
the
operon. How that works, well again, is that's cross. We've got our promoters, our repressor, our operators, and our genes. But if we have tryptophan present, tryptophan would be these yellow hexagons. If we have tryptophan, tryptophan is going to fit inside the repressor, and it's going to change its shape so that it fits in the operator. So in this case, if we have a bunch of tryptophan present, and we don't want to make tryptophan, so the repressor is going to set that operator in position. What happens if all of a sudden
when the ribosome reads it indicates that the protein building is finished.
DNA makes up genes.
So the result of translation is that you built an influence
to make
amino
up different traits
that were brought in certain sequences based on the coding of the
deletion.
We got appreciation of how that happens. We got appreciation of how to use these translation tables, but also how either a point mutation or a frame shift
tryptophan goes away. If there's no tryptophan in the diet, well, the bacteria is not out of luck because it changes now the shape of that repressor. And so once it's changed the shape of the repressor, RNA polymerase can grab on, drive down that operon, make all of those genes, and those genes can be used to create more tryptophan.
We talked about
and so it's a great kind of a feedback loop. So in the lac operon, we're going to turn it on if we want to break down lactose. In the trip operon, what we're going to do is create tryptophan.
Have observed and documented hundreds of mutations in
and I really didn't understand how operons worked until I stopped thinking like a scientist and really started thinking like an engineer. We've got a problem that we have to solve, and one of the great ways we can do that is using an operon. And so I'm going to show you a quick simulation of how this works. It's a PHET simulation. Here's a website. I'll put a link to the simulation down in the video description down below. But it's a great way when I've used it with my students for them to wrap their head around how an operon works. And so we're looking at a lac operon, and to make it simple, we're only dealing with one gene, one of those three genes.
Another fascinating topic for.
And so to look at the simulation,
well,
you could try to figure out what everything is, but it's easier if I show you the legend and show you what everything is. And so we've got RNA polymerase kind of bouncing around here. Again, they'd be moving around. There's molecular motion. They'd just be randomly bouncing around. But this is one of the players. We've got the RNA polymerase right here. We could look down below, and I could ask you, where is the operon? Is this the operon here, or is this the operon? Well, the right answer is this. This is going to be the operon here because it's got a promoter. So let's put that promoter out here. It's got an operator, an on-off switch, and then it's going to have a gene, in this case the LAC-Z gene. So what's this up here? Well, this is not an operon, but this is going to be another what we call a regulatory sequence.
The answer.
And so this one down here is going to make an enzyme. It's going to break down lactose. But what's this RNA polymerase up here going to make? Well, it's coding, it's making a little bit of messenger RNA. But what it's eventually going to make is it's going to make a repressor. Okay, so now we've got the fourth part.
The answer is similar to the last answer, but there are uracil bases
here.
here.
Promoter right here. We've got the repressor. We've got the operator. Now we've got the gene. You can see that the RNA polymerase can't make that gene because the repressor is blocking its way.
DNA
Some dog breeds, however, have extremely long
and that structure will help you understand how
fur.
it's first
By carefully studying the genetic code of over 700 dogs,
so instead of
and then
A
comparing
with
the
T,
DNA
it
to
was
that of the
also
wood,
stressful because G,
I'm a bit of a
scientists discovered a single
but a
point
bit of a
mutation
messy, unable
on
to
a gene
decorate my
called
classroom kind of perfectionist.
FGF5.
Actually, maybe I'm not a perfectionist.
This point mutation,
So let's
which
start looking at a short sequence of DNA.
And I really want myself to
And the letters I'm going to use, these are the shorthands for the various nucleotide bases that make up a
It's a type of
sequence.
nucleic acid
colony.
happens
So
in
it's an
off position.
nucleic acids are
And it should be.
biomolecules.
In other words, we shouldn't start making those genes
Adenine,
until lactose is present. So let's add a bunch of lactose. I'm going to add a bunch of
So that would be our
lactose.
sequence of DNA. And what would be
Add a bunch of lactose, and we can speed up the simulation a little bit.
Sugar phosphate backbone.
What's going to happen is the repressor,
the corresponding sequence of
the
the RNA that
repressor is now having the lactose bind to it,
Remember
changing its shape or it's changing its concentration.
That means
Well, the key thing to appreciate is if we're talking about base pairs in DNA,
well, I'm big on
adenine pairs with
so anytime I would
thymine,
teach biology
cytosine pairs
concepts,
with
I found
guanine.
some ways
But if we're talking about pairing into RNA, well then instead of thymine and RNA, you would have uracil. So the
Once the
RNA
lactose, and
here
you can see it's holding
Thymine
two
and
cases, once the lactose binds to the repressor, it
RNA
can't bind to the operator. And if we play the video, what's going to happen? Well, now RNA polymerase can drive down the operator, down the operator, which produce a protein. In this case, that's going to be a lactose protein. What it's going to do is going to digest and break down all of the lactose.
Everything that's spread afterwards regardless of whether it's really messed up,
from DNA, RNA. Now the next step, if we're talking about codon processing, how does this information actually have an effect on the body? We're going to go
see that it's gobbling up all of those lactose. The only lactose left is those
like Greg Cannon, RNA.
What happens eventually, all of the lactose is going to disappear. In other words, we will rotate. What happens now, well, that repressor is going to bind to the operator again. And now we can't make that repressor. So it's a wonderful feedback. Feedback loop feels a bunch of lactose. Now, because E. coli would be silly to make all of the proteins to break it down. We tell we've got a bunch of lactose. So that's an operon. What is it again? It's all of the genes and then a way to control those genes in one tidy little package.
He inherited his DNA from his mother, and that's when a fragment. I can't know for sure what Spike's looked like, but I do know that Spike inherited his DNA from them. One fun fact, but many snake species reproduce asexually. Had that been the case for Spike, he would have inherited all of his DNA from only one parent. But it would still be DNA.
You can take the thing and copy it.
Just like Spike, your DNA.
Hi, it's Mr. Anderson, and in this podcast, I'm going to talk about transcription and translation. And in biology, that's how we go from DNA to RNA, and then from RNA to proteins. And so we are made up of proteins. In other words, we eat food, and that food is going to be reassembled to make us. In other words, you are made up of proteins, and those were made up of amino acids. And so just like when you start to cook pizza, and if you want to cook pizza, you're basically going to have to get all the ingredients together, and then put those together in the right order to make a pizza. Same thing works inside you. The DNA is going to tell your body how to organize the amino acids into those proteins, and those are eventually
And that structure will help
so if we talk about that cooking analogy, basically, your cookbook, if we're cooking in the kitchen, is going to contain a bunch of recipes to make a number of different ingredients. The chef is going to pull the right recipe out, or you could copy it down on a little index. And then you bring it to the kitchen, you take all these ingredients, and you put them together correctly to make. So if we switch to the next slide, what's been replaced in our kitchen? Well, the cookbook is going to be like the DNA. The nucleus is protected. The chef has been replaced by a number of different recipes.
Not all of your
The next codon is UGA.
This RNA is doing this
very helpful trying to make more cells. One of them is
And then the next one is
until just
sometimes
UAA. Well, here they have this little black circular dot. What does that
But the
mean?
most important
Well, that
part
means
of the
stop
type is the
codon.
base.
And sometimes they'll just write the word stop.
Rich Jorgensen was working at a
stop.
biotech
There is
startup.
not an amino
Recipes
acid
now,
called
messenger
stop. This
RNA.
actually
And if I were to do this
signals
accurately,
this
I
is
should have
happening in the
hundreds and hundreds of
ribosome.
different
This is
ones.
signaling for
One
the
copy of the
translation
DNA
process
doesn't
to stop.
stop.
This is
Hundreds
the
of
end
copies
of our amino acids.
What are the ingredients then? Those are going to be the amino acids, and we're going to put those together in the correct order, and then we're going to work proteins in that
Just like
protein.
These are first.
But now let's do some interesting.
This process that I described,
There are where some of
the
those
central dogma was
bases
developed by this man, Francis Crick. So after Watson and Crick developed the structure of electron, he spent years working out how it works. And so the process he called central dogma. Basically, what we do is take the DNA is going to have sections which are called genes. And the gene is going to code for a specific protein. And so basically,
He added more purple
Or,
recipes.
and there's a It may not be transcribed at all. This is a telling your eye cells don't always have portion of the other is don't say replication. There is regulation.
What would it be? It'll now code for
and so basically, we'll copy down that gene into the messenger RNA. That process is called transcription. Transcription is going to take place in the nucleus.
see more solution. For example, if the gene was swapped out for
Just
A, this gene and then what was going on?
Sam's vision has never been great, and he was having trouble seeing anything.
All of this up here is going to take place in
That
the
routine visit to the eye doctor revealed the problem.
and I'm talking about eukaryotic cells.
The actual translation
Basically, mutations are simply changes in DNA. We'll manipulate changes in DNA bad. Well, DNA basically, once we've done that, then that messenger RNA is going to move out through one of these nuclear pores. It'll grab onto a ribosome, and then it's going to make these
1,000 codons before the end, but all of a sudden you had a
That
point
process
mutation
is called
that would significantly affect the protein. Now another type of mutation that typically has a fairly significant effect is a frame shift mutation. And that's where something gets inserted and or deleted.
So basically, the way I remember it is script in transcription stands for to write. And so we're writing down the message of the gene into messenger RNA. And so it's a message. And then that messenger RNA is going to go out here, and we're going to have a number of amino acids that weave together, and then we're eventually going to create something like this, protein. Now what does that protein make? That protein makes, in other words, you're made up of a bunch of proteins. Where's the recipe book for the proteins inside? That's going to be in the DNA in the nucleus of every DNA. Okay, so let's get more specifically. If we're talking about eukaryotic cells, remember all of our DNA is in a chromosome. And so that chromosome is a bunch of DNA that's wadded up or wrapped around histones. If we unwind it, we can see it's going to be transcribed to make something called this, messenger RNA. And that's going to be translated to make something called a protein. And that protein is going to make, if we really unwind, so if you have changes in the DNA, that's going to cause changes in the messenger RNA, and that's eventually going to change the proteins, and it's eventually going to DNA. It's going to copy the information in the DNA. And so if we've got a C here, that's going to be a G here.
It's the photoreceptors in our retinas that absorb light.
Here we have isoleucine. Which is different than what we had before. We don't have this
complementary bonding that's going to create the transcript. It has the same information as the information that's and then we're eventually going to lose that RNA. Now the nice thing about DNA is it can zip together because there will be hydrogen bonds between the two sides. And now that gene is protected with DNA instructions. So if we go through that in the steps of transcription, this would be RNA polymerase. RNA polymerase is going to grab onto the DNA. There's a number of transcription factors that allow that to occur. Here's our gene. Then as the RNA polymerase is going to move down that DNA, you can see that it's creating a copy. And the copy is called messenger RNA.
So you can
This is our messenger RNA. And if you look, as RNA polymerase takes off, we've left the message behind, and the DNA zips back
into the layer of the transcribed and then translated. Now, lucky for us, even though mutations are always going to initiate, there are many initiating mechanisms to make them less frequent.
Now in us, we're going to do some things too. We're actually going to put a 5' cap on one side. We're going to put a poly A tail, which is just a traditional row. Another thing that we'll do inside us is introns. Introns are sections of that so we'll get rid of those and then splice together.
May no longer be a functional protein because the amino acids, the coding got stopped short. There was a frame shift mutation. So sometimes it could be very bad.
Hi, it's Mr. Anderson. Welcome to AP Biology Lab 6 Truth. This is on molecular biology that must be studied. This material, deoxyribonucleic acid, that genetic material that makes life possible there. That's also in this lab. There are actually 2 parts to it. There's the transformation part where we can transform a bacteria, make it different using a plasmid, and then gel electrophoresis, how we can separate DNA, how we can digest it, break it into fragments, and then separate it according to the length of those fragments. And so messenger RNA. The 2 things we'll do is bacterial transformation. Transformation is when you're taking one bacteria, protein, it's going to have 5 parts to it. It's going to have another wall segment, and we're transforming this by passing DNA to the messenger. In this case, we'll pass a little bit of DNA called a plasmid from one to another. The plasmid we'll be using is something called P-glow. So it's a way to actually get this bacteria to glow, fluorescent glow. Second thing we'll do is gel electrophoresis. cell electrophoresis is separating DNA according to its fragment. Every 3 letters, basically running DNA through a gel. It's almost like jellies and Y's, and then we're separating letters and the length of 4 letters. So those are the 2 things that we're doing. The bacterial messenger RNA. Every 3
attached to the replicator to produce an endless variety of larger proteins.
So mRNA cannot sometimes it feels like DNA gets all the credit. Yes, the structure of DNA is amazing. It has a beautiful double helix structure, a twisted ladder. It has its own emotions. Your stomach cells DNA stores genetic information and codes for your traits. However, sometimes what gets left out is how important RNA. But it's surprising because without RNA, you actually can't get the genetic message out to your cell. But it's not that they can start producing proteins. We talk about this in
and it helps our enzyme figure out where to bind to the strand.
Remember
from our episode about DNA
just as
structure, the
important
DNA strands
DNA.
run in one
In fact,
of
RNA
two
is even
directions,
hypothesized
depending on
as
which
coming
end of the strand is
before
free,
DNA.
and which end
In the RNA world, RNA
one direction is
binds
5
to
and
the
3 primes, and the other direction
So let's
is
make
5. In this case, it's upstream means
with
towards the
RNA.
3
each have
prime
to be
and
formed in
down
a
You will
perfect
find
shape
DNA and
or
RNA in
perfect
all
shape.
the
So the first
important
enzyme
They're
in
the
the
wrong
process
shape.
is
In
RNA.
eukaryotic cells,
Every 3 letters are going to code for one specific
you can find
amino
RNA
acid.
both
The reason it's
inside and
every 3
outside of
letters
the
is it gives
nucleus.
us enough variety
Prokaryotic
to
cells
cover all
don't
the
have
What are
messages.
essentially molecules
Both
that are
DNA and
so
RNA
You'll
are
notice that
nucleic
sometimes
acids.
the DNA
Bacterial transformation we're doing is using a plasmid. This is the plasmid right here. It's called peak low. Basically, peak low is a section of DNA. So in a bacteria, they're going to have their regular DNA, but they're also going to have these little bits called plasmids. Plasmids give them ability to do something. Another type, fertility plasmid to be an example, or resistant plasmid. So that's going to be one that gives bacteria antibiotic resistance. And so in this plasmid, it's been engineered. In other words, it's been built by humans on one bit of the plasmid. We're going to have the origin of replication. That's where the ribosome. So we can attach. These are also going to have ampicillin resistance. So when we grow them on ampicillin, only the ones that have picked up the plasmid are going to grow. Here we've got the glowing fluorescent protein. This is extracted from a jellyfish. And then they also have a trigger, a arabinose sugar. If you add arabinose sugar, they're going to make that glowing fluorescent protein. If there's no arabinose amino acid, it's not then going to flow out into the cytoplasm to grab the next amino acid. Now each tRNA is going to grab a specific amino acid, and it's going to have 3 letters on the bottom of it. We call that the anticodon. It's going to bond to the codon. This here, the messenger RNA.
And the sugar in RNA
It shows us that the steps of the ladder are made up of
and if
4
you
different kinds of chemicals shown here by
deoxyribonucleic acid.
letters.
And so basically this will look over and over until it gets to a stop sequence.
From top to bottom,
So I'm going to start this animation
a
right
single
here.
strand of
So
DNA
basically, let me kind of talk you through it.
Well, if you have a lot of
This
something
down here is going to be the small subunit. This on top is going to be the large subunit. And then we can see that we have all of these, they're blue, these dark blue, are coming in and they're actually bringing their amino acids with them. Now there's an enzyme that attaches that on. But basically these dark blue tRNAs are each dropping off an amino acid, and then they're taking off into the cytoplasm to pick up another amino acid. You can see right here that the messenger RNA that came from the nucleus is just feeding through the middle of that ribosome. And it's a site where we can actually have the tRNA come in. Now if we watch this animation here, this is a ribosome that's bound to the endoplasmic reticulum. And so it's going to attach to the endoplasmic reticulum, and that growing protein that was growing inside that large subunit is actually going to grow into the middle of this endoplasmic reticulum. So we can modify or do things. And so basically this is translation. So translation, if we go back to our analogy again as a cooking analogy, basically the ribosome serves the purpose of the chef. And so it's taking together all these different ingredients, all these different amino acids, and putting it together as a protein.
Instead of apples in the tree,
And
maybe
that
the
wouldn't
apples
just
are
be a waste of resources. That would actually be very difficult to explain to your friends. You want genes to be regulated, controlled. Remember that genes are made up of DNA. DNA is used to give instructions for the production of proteins in the process of protein synthesis. But an important concept is that there needs to be a method of determining which genes will be turned on and which genes will be turned off.
Finishing touches.
And that's an easy one to remember because the R
And there are many
stands for
ways that
ribosomal.
Car in the garage still works though.
The ribosome is
We
going to
mentioned
build our
earlier that
RNA
DNA
and
codes for your
called
traits, but
translation.
it couldn't do that without
You
RNA.
can find a lot of
In our
proteins
protein
and animations on translation that are just fantastic. We're just going to break down some basics of what's happening.
This is called
In the cytoplasm, if you look at this, you have
To help DNA interact with the
molecules
cytoplasm and convert those amino
tRNA
acids
stands for
into proteins,
transfer
We're
RNA.
translating
special
these
chemicals and
into
centrifuges
protein.
make
You can see the
partial
protein
copies
coming
of
out
the
of this.
DNA code.
Okay, what
These
I'm going to try to
partial
do here
copies
is actually
called
decode
RNA
a
look a
gene.
lot
So
like
we've got
DNA,
gene
but they're
found
shorter,
within this
of
stretch
course,
of
and they're
DNA.
missing
So
one of
this
their
is a
sides.
double
We talk
helix,
about
so we
3
have
different types of
complementary
RNA.
strands. And
And
so
they're
basically what I'm
very
going to do is
important
the
roles.
process of transcription.
mRNA, which
So I'm going to
stands for
go
messenger
from
RNA.
DNA.
mRNA's job is to
It's a
carry a
building block for
message
proteins.
based off of
Since
the
we're
DNA.
making proteins,
DNA,
eukaryotic
messenger
cells,
RNA.
DNA generally
And then I'm going to try to
stays
translate that message, and I'm
mRNA
going to
together
go
build
from
the
messenger
take the
RNA, or
message
through the
to
process of
ribosome.
translation, I'm going to
Ribosomes
go
make
to the
protein.
amino acids
And
of the
RNA
protein.
is actually
Okay, so
the
basically,
main
I'm going to play the role in this first line
type of RNA is
of
called
RNA.
smaller shape and
In the
size.
cytoplasm, RNA polymerase is going to look what's in the DNA and it's going to be complementary RNA. So in the DNA, it goes to T, but when that unwinds, the T will actually go to A, and the messenger RNA goes to A, and it goes to U. Remember, we have uracil messenger RNA.
can actually
C, so it goes to G. So if I just keep going, this right there would be
It may not be transcribed at all.
Ribosomes and proteins
Your eyes
So
don't
big
read
picture,
the
hopefully you got a better
3 letters
appreciation for how
suck amino acids out of their cells and stick them together in a chain according to
So
their
every 3 letters in this messenger RNA is going to be what we call a codon, because it codes for a specific amino acid.
Those amino acids are joined
This
together.
particular
They make a
method
polypeptide
is found in
We got a
Proteins are
appreciation
made
of how
with
that
one or
happens.
more of
We
these
got a appreciation of how
proteins.
genes
In
get
this proteins
translated.
have tons of different
Also,
roles.
how
And we don't want to spoil
other
it, so we're going to talk more in our protein
scientists
codon.
can
Before we
eventually
go,
detect
let's try a little 3-question quiz, shall we? Remember, just pause it after the question so we have time to think. Question 1. If I have 8 DNA nucleotides, how many DNA bases do we have? And also, how many bases
tRNA will eventually be,
the answer?
leave behind
Each
amino
nucleotide,
acid.
regardless
That's
of
the
whether
first
it's a
amino
DNA
acid
or
before
RNA
the
nucleotide,
next codon.
has a phosphate,
It can
sugar,
bind
and a
to
base.
the
Carry this on.
So,
repressor.
If you're
8
wondering
DNA nucleotides would have 8 bases.
tRNA
DNA
down
bases pair like
so
this,
then
and
attach
that's
carrying
4
amino
DNA
acid
bases.
Let's
in
talk
the
about
opera.
some key players.
Question
in
2.
the
for that,
If one strand of
Well, then the
DNA has
RNA
reduced
is no
bases,
longer
shown
used.
here,
First,
use
RNA
ATT,
polymerase
determines
G,
which
It's
amino
usually
acid
very
A
very
builder
very
enzyme
like complementary
RNA
DNA
We're not even
bases
necessarily
before
feedback.
the other
If you're in an
enzymes.
environment with
A
lot
lot of
tryptophan,
enzymes
don't
end
create
in that
which
tryptophan.
amino acid
Or if you just have a lot of
RNA
tryptophan laying
codons.
around, don't
I
create
used to have a
more
codon
tryptophan.
chart poster
If
and
you
just
don't have
marvel at
tryptophan
that.
around,
You can
well, then
see on a
the
codon chart that the
repressor won't be
AUG codon
co-repressed,
on the
I guess.
mRNA.
Now, if we want to
And
do
then
the
the
actual
code
translation and figure out what amino
AUG
acids
is
are
all-
created,
So, quick review of
well,
our
I can't
Hot
do
Pocket
this from
Mission
memory. I have to
Impossible
use
paper
one of
so
these
far.
standard
Broken into there
genetic code
containing the
decoders. Some of them will be
instructions,
circular,
copied
some of
down
them will be
those
like this. But
instructions,
basically what you do is
added
you look
some
at the
protective
codon
coatings, and then
in the
cut
messenger
out some
RNA,
extra
so
notes that you
AUG,
escaped
and then I just
back
find
out
that
of the
down here.
layer.
So here's
Now I have
AUG
to
right
actually
here,
read
so
the
that's going to
notes,
be
make machinery,
methionine.
and
So
assemble the
methionine is going to be the
ingredients.
first
This
amino
process
acid.
is called
Now,
translation.
methionine
So
is
next,
special because it
rewind
also
your
starts
memory or
all
just
genes.
watch
All genes are going to
also
start
the
with
start
methionine, and
button.
sometimes
Methionine
we
is
trim that off
probably
later,
going
but
to be
basically
your
that's going to be
RNA
our start
particular
sequence.
needed in
I
order
hope
to
There are
so.
many
Those
types
little
of
dots
amino
on the
acids in the
membranes are
codon
the
chart.
Remember
ribosomes.
that
But there
transcription
are even
Process
more
messenger
possible
RNA
codon
is fed into
combinations.
a ribosome like a
Proteins.
dollar
That
bill
means
into a
there can be
balloon.
more than
Ribosomes
one
are a mixture
codon
of protein
that
and
codes for
second
the
kind
same
of
amino
RNA called
acid.
ribosomal
For
RNA
example,
or
according to the
rRNA. And
mRNA
together as a
codon
sort of
chart,
workspace,
all of these
RNA doesn't
mRNA
contribute any
codons
genetic
here
information
code for
to
the same
process.
amino
Instead, it
acid,
has binding sites that allow the incoming mRNA
That means
to
their
interact with
complementary
another special type
tRNAs
of
all
RNA,
carry the
third
same amino
in this
acid,
paper called
leucine.
transfer RNA or
Okay, so going back
tRNA.
to the
tRNA
mRNA,
really might as well
let's
be
try
called
the next
translation
codon
RNA
on
because
this
that's what it
mRNA.
does. It translates from the language of nucleotides
On the
into
codon
the
chart, you can
language
see
of
that
amino
codes for the
acids and
amino
proteins.
acid
On one
proline. The complementary tRNA has the anticodon GGU. And look, there's
Basically, what we'll do is we'll have 4 different types of bacteria. The bacteria we're using is E. coli, Escherichia coli. So basically, we're going to take some of the E. coli, and we're not going to give it the plasmid, so it gets no DNA.
As the chain grows,
we'll
it
place
bends,
some of that
it
on what's called
folds, and it
Loria
sticks to
broth.
itself
That's just basically
to form
food
a
for
perfectly
bacteria.
shaped
And then we'll
protein.
plate some of it on Loria broth with ampicillin. So since it has an antibiotic, it's not going to be able to grow. We'll then add the plasmid to some of the bacteria. We'll grow some of them on Laurier broth and ampicillin. Some will be able to grow, those that pick up next plasmid. And then we're also going to have some of those with this trigger, this arabinosugar as well. And I'll show you what all those things are going to look like in just the next one. First, let's discuss it. So basically what you need, that's glutamic acid. So that's going to be
once a protein is built,
you go to the next one, it's CAA.
If you watched our DNA replication video,
The answer. So remember those base
that's
pairing
glutamine, so that's going to be
and the popular
GN.
mnemonic.
Then our last
Placing
one
the
is
bases
going to
in
be
this image,
UAA.
reading
So
in
when
the
I find
direction
that,
of the
it's
proline,
going to
they
be
mean
UAA. It's going to be a stop sequence.
transfer
What does
that
that mean?
Question 3.
Well, that's the
In a
end of the
positive
gene,
way,
so that's not going to actually code for an amino acid. We'll actually put in a release factor, so the whole thing can break out.
And
So
more
this
about
is going to be our
that.
first
There's
amino
this other
acid, the
sequence
second
here.
amino
If I
acid,
still
the
had the
third
original
amino
portion of
acid.
DNA
the
here,
operator
what would be
start
the
the
complementary
something
RNA base?
called
But
a
when the ribosome
repressor
reaches it,
enzyme.
it indicates
Some of them
the
on
repressor
Laurier broth
is
and
bound to the
ampicillin.
operator.
So the
Some will
results
be able to
RNA
grow, those
that
that pick up the plasmid,
Or
and then we're
RNA
also
not
going to have some of
forward.
those with this
No
trigger,
mRNA
this
is
arabinosugar
based
as
on
well. And I'll
coding
show you what all those things are going to look
The
like in
answer.
just a second.
So remember,
But first,
it's
let's
asking
discuss
for
it.
RNA.
So basically,
no thymine here. It's uracil instead. Apples under helps you remember A with U. Car in the garage
So to
helps
answer
you
the
remember C
original
with G.
question,
So
what
in this
is
portion,
DNA?
reading in the direction shown on this
DNA
image,
is a
we'd have
molecular
these
the
RNA
fourth
bases.
amino
for a
acid. This
living.
whole
Notice
thing then
the
is called a
answer is similar to the
polypeptide.
last answer,
And
but
again,
there
it
are
would
uracil
fold
bases
into
instead
a
of RNA
specific
creates
protein to do
One
a
last
specific
thing.
job.
Our
Now,
models
most of them are going to
here,
be much
they're unable to
longer
show
than this,
the
but what I've shown you is how
shape
DNA
life.
becomes messenger RNA. Amino acids will eventually form proteins, and now that whole thing is eventually going to make you, because you're made up of proteins. And so that's transcription, that's translation, and I hope that's helpful.
One end of the tRNA is an amino acid. On the other end is a specific sequence of 3 nitrogenous bases. These two ends are kind of matched to each other. Each of the 20 amino acids that we have in our body has its own sequence at the end. So if the tRNA
of the
has the amino acid
mRNA.
methionine
But remember,
on one end,
that
for
mRNA
instance,
was
it can have
complementary to the
UAC
DNA.
as the
So the
nucleotide
DNA
sequence on the other
ultimately
end. Now,
was the
it's just like
director of the
building a
entire
puzzle.
So let's take a look at our
mRNA
setup
slides
here.
through the ribosome.
This
couldn't have done it
example
without
called
some serious help
lactose.
from
Three letters
mRNA.
behind.
Notice there's a
RNA,
promoter region
and
of DNA.
Codon.
The
ribosome then
operator
finds
region
the
of the
matching
DNA,
piece of the
and there
puzzle
are
of
3
tRNA
genes
with 3
here that
bases
code
that
for
will pair with the
enzymes that
codon
help
sequence.
in the
That end of the
process of
tRNA, by the
breaking
way,
down
is called the
lactose.
anticodon. Sorry for all the
Lactose
terminology.
is a sugar.
You need to
If
know it.
lactose
And of
sugar
course,
is
by
around,
bringing in the matching
bacteria
tRNA,
want
they're
these
also
enzymes to be
bringing
made
in
so
whatever
that they can
amino
use them
acid
to break
on
down
the
the lactose
tRNA.
sugar.
Okay, so starting at the
Then they can
top line,
metabolize it.
All very
Fed
fast.
bacteria
Hi, it's Mr. Anderson, and in this video I'm going to talk about
Here's the
the
Another
repressor.
fascinating
The operon was
There's
discovered in the
actually
1960s by
codes for
3
producing
French
repressors.
scientists,
See this gene that we call
Roux
tRNA?
and Monod.
It has its own promoter.
And on the
Well,
other end of
this
that
gene
tRNA
codes for the
is methionine.
production
The
of the
mRNA,
repressor.
like a mile-long
So
dollar
why do
bill,
we need
keeps
this
sliding into the
repressor?
ribosome so that the next
Well,
codon
it's
can be
wasteful to
read,
make things that
and
you
another
don't need.
tRNA
If there's no
molecule, the right
lactose, it
anticodon
wouldn't make
binds
sense
on.
to
If
start
the codon
making
is
enzymes that
UUA,
work together
then
to
the
break
matching
down
tRNA has
lactose.
an AAU
It would be a
on one
waste.
end and
The
a
enzymes
leucine
would
on
just
the
sit
other
there.
end of the
Really
mRNA.
bad
So if
French.
lactose is not
But they got to
present,
coin the term
then the
operon, and
repressor
basically it
binds to the
means to
operator.
operate.
And this
And so it's
blocks
mostly
RNA
found in
polymerase.
basically what you get is a
mRNA
plasmid.
that
The plasmid is a little bit of
And therefore
DNA.
proteins,
We're going to add it to
in
general.
bacteria.
It's just so the protocol for doing that basically entails
it led to other terms that you maybe
entails
never
putting
heard
them
of
on
before,
ice,
like
the bacteria.
regulons and
You then add a
modulons and
transforming liquid.
stimulons
It's
are
calcium
groups of
chloride is what it
operons
is, and that
operating
causes
together,
them to kind of
from
loosen up their
one
membrane and more
stimulant.
likely take in
And so
that,
it just means to
plasmid.
operate. That's
We're
what
also
an
going to heat
operon
shock them,
means.
so they'll be on
And
ice,
so
and we'll heat
the
shock
one
them.
that they
and
discovered
some of
was
them will
the
pick it up. And when I do this in
lac operon. We're
class,
studying this in
you know,
E.
it's
coli.
2 or 3 groups
And so the
out
lac
of
operon
20
is
or 30
designed
groups
for E. coli
will actually get it to
break
transform.
down
So there's a lot of
lactose.
chance to that. But if you get one bacteria to pick it up, you can get a colony of that, and then it's going to grow. And so basically, let's kind of talk through it. If bacteria doesn't pick up the DNA, what should it look like here? Well, we should have a So basically what we'll do is we'll have 4 different types of bacteria. The bacteria we're using is E. coli.
Remember, if your mutations are simply
so basically we're going to take some of the E. coli.
Why is
changes in DNA not going to DNA, remember, no DNA. We'll plate some of that on what's called Loria broth. That's just basically food for bacteria. And then we'll plate some of it on Loria broth with ampicillin. So since it has an antibiotic, it's not going to be transcribed and add the plasmid to some of the bacteria.
This is a messenger RNA, and that's going to be translated to make something called a protein. And that protein, that's not how you spell the plasmid. So if you have changed the way that I remember, that's going to cause changes in the messenger RNA, and that's eventually going to change the proteins, and it's eventually going to change.
First, let's discuss
you can remember.
So basically,
transcribes
it's going to get you half of the way there to
from the
remembering
genes on the operon.
words. And so the neat thing about bacteria is that if they have a number of different genes that are used to achieve a certain task, they'll put them right next to each other. And so as the RNA polymerase moves down the DNA and makes all of the messenger RNA and makes all the proteins, you're just ready to do whatever the job is. And so the genes are going to be located right next to each other. In this case,
ampicillin, we should have no growth here because unless they get that ampicillin resistance, they're not going to be able to grow.
The promoter is going to be the region of the DNA
ampicillin,
where
the ones who get the plasmid, should be able to grow if they're transformed, and then these are going to be the only ones that actually glow. And so if we plate them on it, what we find is that the ones that actually didn't get the plasmid, but they're on Loria broth, what you'll get is they'll cover the whole of the plate. You can see that on here. The ones who we try to grow on ampicillin, the ampicillin is going to kill them. In other words, it's going to kill all of them since none of them picked it up. Likewise, on the ones who have the LB and the ampicillin, the ones that are transformed, we're going to see some colonies. So each of these colonies represent one bacteria that picked up the plasmid. It's able to make copies of itself. It's able to grow. So you can see not a ton of them were able to do that. But then finally we get the really cool plate where we've got LB ampicillin. So all these bacteria were transformed, but we also have that arabinose sugar. So they're able to use that to produce this glowing protein. And so you get this fluorescence in the bacteria. And so what do you need to understand? What a plasmid is, how it transforms bacteria, and then how that might be manifested in all the different growth on all the different plates. Second part of the lab is called gel electrophoresis. Gel electrophoresis is going to use a gel box that looks like this. There'll actually be liquid in it. And then you'll have a little fragment of jello. This is an agarose gel. So this is what it would look like. You're going to load up the DNA in these wells on one side, and then they're going to migrate across. They're going to be pulled, since they have a negative charge, they're going to be pulled towards the positive end. And so we let it run for a certain amount of time. Now, if we were to let just DNA run, what you'd get is one fragment being pulled across. The smaller fragments of DNA are going to be able to make it farther.
But mutations are also good.
The smaller it is,
all life on our planet started with a change in the DNA that was selected for or against, creating wonderful diversity.
But we also want to trim the DNA, so we use something called a restriction enzyme. Restriction enzymes come from proteins. A good analogy is to think about recipes. Constantly, this is one of my favorite recipes. They use restriction enzymes to cut up all their DNA, remove the viral DNA, and so basically what I'm going to do is I'm going to make a restriction enzyme is going to cut between
carefully, see if you can
the nucleotides. So it's going to cut the DNA in specific points, but it's only going to cut the DNA when it has this specific reading frame. Basically what you get is a plasmid. The plasmid is a little bit of DNA has been changed to we're going to add it to bacteria. And so the protocol for doing this is suggest it using a restriction enzyme, and that's going to cut the DNA up the bacteria a little bit. Then if we were to use a different restriction enzyme, it would liquid it into calcium chloride is what it is, and that causes them to run all of this through a gel. Well, this would be one big fragment, so this would not move very far through the ice. Then we'll heat shock. This fragment way up here is very small, and so when I do this in class, it's 2 or 3 groups out of 20 or 30 groups, we'll actually get it to transform. So this would be how this DNA would separate. But if you get one bacteria, pick it up, you can get a colony of that, and then it's going to grow. And so basically, let's kind of talk through it. If bacteria doesn't pick up the DNA, what should it look like here? Well, we should have a and so it would be migrating even farther. And then these ones would be, I don't know, somewhere in the middle. something like that. So this would be how this DNA would separate. If we were to use a different restriction enzyme, we'd cut it up differently. Now,
an extra gene for purple, you should not get more purple, but less purple. It took a decade of brilliant scientists working on petunias and fruit flies and worms and other organisms to finally work out what was going on.
That mRNA will be used to make enzymes to break down the lactose sugar. And bacteria like to eat, so that makes them pretty happy. We have to say that we think it's pretty impressive to think about all the gene regulation that occurs in cells. And if you find it fascinating, know that there are careers that focus on gene regulation.
But this fragment way up here is very small, and so it would be migrated even farther. And then these ones would be...
Because by doing that, we can gain a better understanding of a variety of
So this would be how
diseases
this
that have
DNA
gene
would separate.
influences
If we were to
in the
use a
body.
different restriction enzyme, we'd cut it up
That's
differently.
it for the Amoeba Sisters, and we remind you to stay curious.
You might call Stone a gene doctor. There are many causes of blindness, but RP is an inherited disease.
Well, we should have a lawn of bacteria.
It is caused by a change
Ampicillin, we should have
or a
no growth
mutation
here
in a
because
gene.
unless they get that ampicillin resistance, they're not going to be able to grow. LB ampicillin, the ones who get the plasmid, should be able to grow if they're transformed, and then these are going to be the only ones that actually glow.
Signals to the brain.
And so if we plate them on it, what we find is that the ones that actually
as more photoreceptors
didn't get the plasmid, but they're on loribaroth, what you'll get is they'll cover the whole of the plate. You can kind of see that on here. The ones who we try to grow on ampicillin, the ampicillin is going to kill them. In other words, it's going to kill all of them since none of them picked it up. Likewise, on the ones who have the LB and the ampicillin, the ones that are transformed, we're going to see some colonies. So each of these colonies represent one bacteria that picks up the plasmid. It's able to make copies of itself and it's able to grow. So what do we try to break down? But then finally we get the really cool part where we've got LB ampicillin, so all these bacteria model right here. We also have amounts of DNA, so we use micropipetters to produce this glowing tiny amounts of DNA.
All of a sudden, a lot of the
essentially loaded in here.
What is the lactose?
So what do you need to understand what a plasmid is? Let me kind of quickly talk about analysis. So none of that light beam moving around running a gel is eventually a ladder. Or second part of the ladder is called gel electrophoresis. Gel electrophoresis is engineered into the repressor. Sent to you. Each of those fragments is going to have a different length. So this would be a 200 base pair, 300 base pair. And then you'll have a little fragment of 400 base pair. So these are known quantities of DNA. So this would be the ladder on one DNA and these wells on one side, and then another migrate. So we can see that this one right here, cold, since they have a negative charge, interpolate between these two that this is maybe 200. So we let it run for a certain amount of nucleotides between it. Or you could look over here. Here's another gel. So we loaded it right here in the wells. It's moved in this direction towards the positive end. You can see here's our ladder on this side. But you can see that we cut it with a different restriction enzyme. In this case, we're using a plasmid. So that's just that little section of DNA. We use different restriction enzymes. So it's the same DNA, but by using different restriction enzymes, this one looks like a BRCA2, this is maybe
quick digression. Everything motor, operator, and gene.
So basically, we can do analysis. We can compare that DNA to DNA ladder.
The repressor is a little bit different.
That's it. So that's molecular biology. Just DNA run. What you get is one fragment being pulled across. The smaller fragments of DNA are going to be able to make it farther, and so the smaller it is, the farther it's going to migrate. So biology, this gel right here is a smaller fragment of DNA than this one is. But we also want to trim the DNA, and so we use something called a restriction enzyme. Restriction enzymes come from bacteria. Basically, bacteria are constantly being infected by viruses, so they use restriction enzymes to cut up all their DNA, remove the viral DNA, and then glue it back together again. And so basically, a restriction enzyme is going to cut between the nucleotides. So it's going to cut the DNA
another point
in specific points. It's only going to cut the DNA when it has this specific reading frame right here.
There's going to be a regulatory sequence.
What I've done is I've changed the number of T's beyond the direction 1. So basically we can digest it using a restriction enzyme going to taste like now. They're probably going to off a little bit. Way too much. To use a different restriction enzyme. What is that? They're going to taste a smaller bit. Opens up in a region where that promoter runs. So let's look at another mutation. This be one RNA polymerase, so this is going to drive right down the DNA. It's going to make this fragment RNA up here is very small, and so it would be migrated. What are the proteins designed to deal with? Lactose. And so they're going to break down the lactose. Now all the lactose is gone. What's going to happen to that repressor? To use a different restriction enzyme. Again, the first two are just changes in one letter into the let's look at this mutation. So if there's no lactose present, we would say, again, what are we doing? Well, basically we're shifting this to turn on the oven to the end instead of where it should be, sitting inside the operator. Is that going to change our cookies? For sure. We're going to end up with not cookies. We're going to end up with dough. And so if you think about this, changes in the DNA is just like changes in the recipe. What causes them, however, it's not a type.
You should not get more purple, but less purple. It took a decade.
We're going to end up with dough. And so if you think about this, changes in the DNA is just like changes in the recipe. What causes them, however, it's not a typo. Basically, there are two ways that you can get mutations. The first are what are called spontaneous mutations. That's when just something in the process of replicating the DNA or forming the gametes goes wrong. And so a very common type, it's not that common, but a common type is called strand slippage. Now if it's in the area where there's no genes, it may not be strand and the daughter strand area, especially an area, and then you get bonding that's not pressing genes. And so that would be an example of a spontaneous mutation. But a lot of mutations are caused by our environment. And so basically what are called induced, just like with the recipe and the cookies. So if we can have mutations just at one point, then we call those point mutations. Or we can have larger mutations where big chunks of the chromosome are being used. And so let's look at the first type of point mutation. So if I go and mutate, and if you cut that, let's try that again. This is called a substitution mutation. So right here we've added a C instead of a T.
To the point that the entire cell is filled up with this. And the cell explodes, releasing these viruses to go and then infect whatever other
so you might
cells
think
they can
that's
find.
bad. Well, we've got protection for that inside our cell. There are going to be, for example, proteins that are going to cruise up and down our DNA. And when we have A bonded to C, or cigarettes, quite match it's a chemical that's caused. So we're going to know that there's something wrong. To know what was going basically on. These enzymes could cut out the C, and then they can know exactly how to work in again. And so recent studies have shown that this chemical is just one of the mutagens. A C on this side, and A on this side. It's called benzoapyrene. Not basically, if you might look at this chain, right, we have to get rid of this right into the double helix, and it's causing the 50% of the time they're going to replace the wrong letter. And if that letter is not inside a gene, it's no big deal. But if it's inside a gene where we're actually making amino acids to make proteins, it can be a huge deal. That's a substitution. We could also have radiation as micro amounts of DNA, so we use one of those micropipetters to make a tiny number of proteins that can fix that as well. Sometimes when they fix it, they'll actually add a new letter. Let me kind of quickly talk through it. You can see it's totally fine. Another thing a lot of times we've added this new letter here on this cell replicator, or a copy of itself. DNA ladder is going to now have an engineered, it's sent to you, and each of those fragments is going to have a different length. So this would be a 200 base pair, 300 base pair, so this would probably be 400 base pair. So these are known quantities of DNA. So this would be the ladder on one side, and then you run your DNA in another length. So we can see that this one right here, you can kind of interpolate between these two that this is maybe 250 base pairs or 250 nucleotides. Or you could look over here. Here's another gel. So we loaded it right here in the well. What that could do is it could shift all the letters positive end. And that gives our ladder on this side. But you can see that we cut it with a different restriction enzyme. Now, if it's in this case where there's no genes, it may not be harmful. But if it's an area, especially if you look at our DNA again, so nothing in the DNA, maybe it's a chemical or radiation that causes using different nucleotides to be lost. So now we're missing something. So basically, now when the DNA tries, just like with the recipe and the cookies, we can have mutations just at one. So basically, we can do analysis. We can compare that larger DNA ladder on the side. Big chunks of the chromosome are being. And so let's look at the first molecular biology mutation. So if I go and mutate, I don't know if you caught that. Let's try that again. This is called a substitution mutation. So right here, we've added a C instead of a down and code for those genes because the repressor is sitting in its. What happens next? Well, lactose shows up again. Lactose would
move into the repressor, free it up, and then it's going to be able to make those genes. And I've got a simulation at the end of this video that shows you in a little more detail how that takes place. Before we get there, I want to talk about the trip operon. The trip operon does essentially the opposite of that. So you might think
of DNA
evolve well in bacteria. We've got protection biology, that's the cell. We're going to specifically to deal with proteins that are going to cruise up and down our DNA. It's going to be an amino acid A bonded to C. It's not going to quite match up those 20 essential. So we're going to know that there's something wrong. So basically these enzymes can cut out the C, and then they can put the T back in again. Where does that message go? We're going to get tripped. But let's say they're cruising down and they see there's a C on this side and A on this side. Well, they might think, what's going to occur there? That they might realize that we have to get rid of this A, and so they get tryptophan, so 50% of the time they're going to replace the wrong letter. High level. And if that letter is not inside a gene, it's no big deal. But if it's inside a gene where we're actually making amino acids, here's our messenger RNA. Huge is going to be our messenger. That's messenger operon here. We could also have radiation or chemicals that are causing the DNA to break apart. Our repressor operator, a number of proteins that can fix that as well. Sometimes when they fix it, they'll actually add a new letter. So this strand you can see is totally fine, but now we've added this new letter here. When this cell replicates, it's going to change its shape. This side, if we copy the DNA, it's fine, but if we try to make copies of this side, we're going to miss present portions, or at least we don't want to make tryptophan. And so the repressor, we can also have major changes, or large-scale changes. And so what happens if all of a sudden it could occur during mitosis, it could occur during meiosis. But basically what we're doing is instead of
isolated DNA from the blood cells. The goal was to find differences in the gene sequences of patients with RP compared to their family members with healthy vision. Those differences identified genes associated with RP. Today, with contributions from other research groups, many mutations have been identified.
Diet. Well, the bacteria is not out of luck because it changes now the shape. It shift all the letters over. So once it's changed the shape of the repressor, RNA polymerase can grab on, instead of losing one letter or one, it could make all of those genes and those genes. If we look at our DNA again, what's another thing that could happen? Well, maybe it's a chemical or radiation deletion. We're missing a huge chunk loss. So now we're using it on thousands. Now when the DNA tries to fix it, hold on it. We still have missing one letters, and the messenger RNA is going to code for. I really didn't understand how operons work. The reason why is stopped thinking like a scientist and started thinking like an engineer. We've got a problem that we have to solve. One of the great ways we could do that is we're talking about this could be a duplication. We're duplicating. I'm going to show you a quick simulation of how this works. We could have an inversion simulation. Here's a website. I'll put a link to the simulation down in the video description. Every 3 letters are going to. It's a great way when I've used it with my students for them to wrap their. The reason it's every 3 letters is it gives us enough. We could also have an insertion where we're taking one chromosome and actually inserting that into another gene, one of those 3 genes that are found. That might seem to look at the simulation crazy, and it's like try to figure out never exist, but it's easier if I show you. But if you think about it, these genes have chromosomes, and so it doesn't mean that that individual is necessarily going to be moving around. This could be during mitosis, and so it wouldn't be a very big deal at all. Or it could occur during meiosis, but we still have these. We could look down below, and so this would be an insertion, and then we could also have a translocation, where we're taking one chunk of this one over here, and this one back. The right answer is this. So there's a ton of different mutations that we can have of chromosomes, and that's probably how chromosomes can operate. Maybe they're on top of another. Some are good, some are called transfer. In this case, the transfer RNA. So what's this up here? Well, this is not an operon, but this is going to be another what we call a regulatory sequence. And so this one down here is going to make an enzyme. It's going to break down lactose. So what's this RNA polymerase? Its job is to transfer amino acids from this site of RNA. It's eventually going to attach, but they're all changes in the DNA. Now we've got some 4 parts to this protein. And so basically, what does a transfer RNA do? Motor right here. We've got the repressor. This side, if we copy the DNA, it's fine. But if we try to make copies of this side, we're going to miss make that one letter because the repressor, then it's going to lock in. And we can also have major changes or large-scale changes. This amino acid, it's then going to occur during mitosis. It could occur during meiosis. Now, basically, what we're doing is instead of specific amino acid, and it's going to have 3 letters on the bottom. An off-position anticodon that's going to bond to the code. In other words, we shouldn't start making those genes. We could lose big portions of the chromosome, all the genes that are found. Add a bunch of lactose. So this would be a deletion where we're missing this huge chunk of lactose. It could be hundreds of speed up thousands of genes. What's going to basically this is the repressor and over and over and over. This could be a duplication where we're now having the lactose bind to it. We could have an inversion where we're copying this, but it's basically going to be kind of upside down. This down here is going to be the small subunit. This on top is going to be the large subunit. And then we can see that we have all of these, they're blue, these dark blue, are coming in and they're actually bringing their amino acids. Once the lactose, and you can see an enzyme that attaches that. Once the lactose, basically these dark, we could also have an insertion where we're taking one chromosome and actually inserting there, taking off the cytoplasm. Now RNA polymerase can drive down the right here that the messenger RNA that came from the nucleus could never exist in the middle of that ribosome. But if you think about it, it's a site genes have chromosomes, and so it doesn't mean that that individual is just all of this is a ribosome. This could be during mitosis, and so it wouldn't be a very big deal. So it could occur during meiosis, but we'd still have these in that growing protein. So this would be an insertion, and then we could also have a translocation where we're taking one chunk of this one over here and this one back. So there's a ton of different mutations that we can have in chromosomes, and that's probably how chromosomes go back to our analogy again. Some are good, some are the cookie analogy. Basically, the ribosome serves the purpose of the shell, and so it's taking together all these different ingredients, all these different amino acids. Some are bad, but they're all changes in the DNA.
So let's imagine that the virus is a pirate ship. It lands. It then sends invaders inside the cell to shower recipes down to those cooks.
Differences identified genes associated with RP. Today, with contributions from other research groups, many mutations have been identified, and close to 100 genes are known to be associated with RP.
These mutations are all very well established disease-causing mutations. There's no question that they caused RP.
And you can see that it's gobbling up all the lactose. The only lactose left is those that are translating into a function. What happens eventually, all of the lactose. Okay, what I'm going to try to do here is actually decode a gene. So we've got gene found in this stretch of DNA. Repressor is going to bind to the operator. We have complementary. Now we can't make that. And so basically what I'm going to do is the process of wonderful feedback. I'm going to go feedback loop. DNA deals with a bunch of lactose. Because E. coli would be silly, must make all of the proteins. And then I'm going to try to translate that message, and I'm going to go from messenger. So that's an operon. What is it again? All of the genes, and then a way to control those genes. Okay, so basically, I'm going to play the role in this first line of RNA polymerase. So in the cytoplasm, RNA polymerase is going to look what's at the DNA, and it's going to put a complementary RNA. So in the DNA, A goes to T, but when that unwinds, the T will actually go to A, and in the messenger word. T goes to A goes to U. Remember, we only have uracil in messenger RNA. G goes to C goes to G. And so if I just keep going, this right here would be an A, and a C, and a G.
She can see flowers
again. This is going to be a G, and an A, and a G. And this is going to be a C, and an A, and an A. And then this is going to be a U, and an A, and an A. And so every 3 letters in this messenger RNA is going to be what we call a
result. I can play bridge now.
Which is very important.
I'm not great, but it's part of my life.
She can see flowers again.
That's still not the end of it, as if to try to confuse me to protect the secret hot pocket recipe.
Did it work?
Marty's vision has improved. It's a very promising result.
I can play bridge now.
The reason Marty has so many blood vessels growing in her eye, clouding her vision, is there's probably a mistake in her DNA, in a gene that produces too many recipes that say, make more blood vessels. So the chefs cook up proteins for more, and she ends up with too many blood vessels. Her doctor wants her to have fewer blood vessels. But how do you get the chefs to make fewer blood vessels?
It's pretty easy. You want to shut down a gene? Put in a copy of the gene with its mirror image. Signals the cell.
which is very
I'm
important.
not great, but
are called the
it's part
exons
of my
because
life.
they will eventually
She can
be
see flowers
expressed.
again.
The junk
some of them are
that gets
just
cut out
gorgeous.
are just the intervening
And
segments
didn't find
or the
anything.
introns.
The most
Material in
likely
the
reason
introns
they
will
did
stay in
not
the
find
nucleus and get
anything
recycled.
was
So,
that
for
Sam had a
instance, titan
mutation
down
in one of the
there is thought
genes
to have
associated with
hundreds
RP,
of exons,
but it
when
was
it's
a
all said
rare
and done, probably
mutation
more than
that hadn't been
360.
So
discovered
apparently
yet.
they
More
did
than any
trick
other
the
protein.
copy in Marty's
And it
cells
also
to
contains
reduce
the
vein
longest
production,
intron
although
in
not
humans,
completely.
some
I see the
17,000
yellow
base pairs long.
inside is
Man,
just
So,
it is just a
Stone
world
took a
record
more
holder.
detailed
Now that
look
it's
at
been
Sam's
protected and
copies
refined,
of
messenger
each of the
RNA
known
can now
genes. So,
move
Stone
out
took
of
a
the
more
nucleus.
detailed
Okay,
look at
so
Sam's copies
yes.
of each of the
Identifies
known
the
genes
problem
in
in
another
RP.
25% of
recipes to the
RP
right
patients. This
cells?
step identifies
But
the
once
problem
again,
in
the
another
treatment
25% of RP patients.
So now we're down to the
But once
last
again,
25%.
he came up
In
empty.
mice,
And
RNAi
that's the
has
situation
now
in
we're down
which
to
the last
Huntington's disease,
25% be a new gene. And that's the
breast
situation
cancer.
in which
So
no
says
one
Greg,
ever
if
knew
we
about.
ever
It's
work
a
this
gene
out
that no
via
one
new
ever knew
genes,
caused
not
retinitis
sort of
pigmentosa
disease that
that
you
no
can
one ever
have.
knew
Stone
about.
was
But a gene that no
determined
one
to
ever knew
identify
caused
that
retinitis
gene.
pigmentosa.
So they looked for about a
Stone
year
was
or so.
determined to
It took them
identify
a lot
that
longer than they
gene.
expected. So they looked for about a
In the
year or
end,
so.
their work
It
pointed
took
to
them a
a
lot
gene
longer
that
than
presented a
expected.
puzzle. In the end, Sam's mutation was in a gene that presented a puzzle for the proper functioning of a molecule. Sam's mutation was in a gene that is required for the proper functioning of a molecule. Cells throughout the body rely on tRNA molecules or tRNA proteins. Cells throughout the body rely on tRNA molecules to produce proteins.
Diseases. Because what RNAi does, remember, the cop's job is to turn off information.
So, a quick review of our Hot Pocket Mission Impossible caper so far. We broke into the layer containing the instructions, we copied down those instructions.
You have 20,000
shorthand,
genes.
we added
How in the world
some
are you supposed
protective
to know what
coatings, and
each
then we
one
cut out
does?
some extra notes that we didn't
one
need, and then
very
we
good
escaped
way to
back
start
out of the
would be
layer.
to look
Now
at
I have to
gene
actually
number
read the
one
notes,
and see
make machinery and assemble
That can
the
be
genes.
possibly tRNA synthesis is so
next,
central
behind your memory, you're
to
just
biology.
watching that
This
video
is so
again. The
central. If
episode
you couldn't do
about
that
animals.
to
Do
biology,
you remember the
then
rough
number one.
endoplasmic reticulum?
If you couldn't do
I
that,
hope you do. Those
gene
little
number
dots on the
one is
membranes are
involved
the
in color
ribosomes.
production.
In the
Try
process,
gene number
messenger
two.
RNA gets fed into a
No
ribosome
petals.
like a dollar
Gene
bill
number
into
two
a vending
involved
machine.
with
Ribosomes
petals,
are a
and
mixture
so
of
on.
protein and a second kind of RNA.
You could make too
called
many leaves, they
ribosomal
could
RNA,
curl
or
up. They could be
rRNA.
upside
And
down.
then
Further
All the
research
bullshit.
showed that Sam was alive because his mutation only reduces,
Instead, it has
but does
binding
not
sites that allow the
eliminate,
incoming
tRNA
mRNA to
function.
interact with another special type of RNA, the third in this caper called transfer RNA or tRNA. And tRNA really might as well
showed
be
that
called
Sam was
translation
alive
gene
because
that's
his
what the
mutation
gene
only
translated
reduces,
language of
but does not
nucleotides
eliminate
into
tRNA function.
Is just
on
amazing.
one end of
10
So
years
maybe
ago, when we were
there was
sitting around
just
talking
something
about
about
what would we really
the
need to understand
demand
the
for
human
protein
genome.
synthesis in
So
the rest. Maybe there was just something about the only tissue demand for protein synthesis in the retina that made that tissue the only tissue that
Stone's
was really
knowledge,
susceptible.
Sam was the only RP patient known to science with that particular mutation. To Stone's knowledge, Sam was the only RP patient known to science with that particular
these two
mutation.
ends are kind of
Sam was
matched.
the only person
asking
that we
amino
had
acids
that had
do
that
I
gene
believe
type.
that enough?
It's not really
To
good
the
thing to be
extent
unique to
that
this
you
case,
were
unfortunately.
replace
amino
that, I was
acid
asking
gene
myself,
on one end,
do
for
I
instance,
believe that
can have
enough? To the
A
extent
unique
that you would
instance is not
replace
strong
that
evidence
gene
that a
with
mutation is
gene therapy?
associated with a
Probably
disease.
not. So what do
A
you do
unique
to
instance
make
is not
yourself
strong
believe
evidence that a
something like
mutation is
that?
associated with a disease.
So what do you do to make all of
that
the
allow
yourself
tRNA
believe
with 3
something?
bases
All
that
this
will pair with
morning
the
was
codon
gone.
sequence.
If
That
you
into
haven't
the
so what
tRNA
you
allows
do is you take all the number of patients out and
You
all
need to
the
know it.
screens
And of
go
course, by
on,
bringing
and now
in
you
the
haven't
matching
found a
tRNA,
specific
also
gene
bringing in
which
whatever
in this
amino
patient,
acid is on that
and you
tRNA.
go
Okay, so
ask
starting at
whether
the
there's
5'
anybody
end
else
of the
that has that.
mRNA that's fed into the ribosome after the 5'
Stone's
end.
painstaking research finally paid
Now you
off.
take your specific gene
He
that you found
identified
in this
two
patient,
brothers with
and you
mutations
go
in the
ask
same
whether
gene
there's
as
anybody
Sam's.
else that
And on
has
the
that.
other end of that
at the moment,
tRNA is
it's only
methionine.
you and
The
this
mRNA
two
Stone's
guys.
painstaking
dollar
research
bill
finally
keeps sliding
paid
into the
off. He identified two brothers with mutations in the same gene as Sam's.
If the codon is UUA,
at the moment,
then the matching
it's only you and
tRNA
two
has
other
a
guys.
U
Now you're up to a
on
whopping
it,
3 patient
and if
generations
the
in this gene. Now are you believing it?
Wanting still more evidence, Stone turned to another powerful tool used in genetic research, a model organism. Stone and his colleague Diane Sluzarski genetically engineered zebrafish to have a mutation in the same gene as Sam's.
Now are you believing it?
To see if it caused RP in the fish. Evidence, Stone turned to another powerful tool used in genetic research, a model organism. Stone and his colleague Diane Sluzarski genetically engineered zebrafish to have a mutation in the same gene as Sam's to see if it caused RP in the fish.
In each case, that new amino acid gets connected onto the previous amino acid, starting a polypeptide chain, which is the very beginning of a protein.
When the engineered zebrafish embryos did not respond to a visual stimulus, Stone was convinced he'd found the source of Sam's RP. Now, he and his team are working on a gene therapy to deliver a fully functioning copy of the mutated gene, fully functioning copy of the mutated gene. Gene therapy has already been used to treat patients with another type of blindness, to treat patients and other genetic blindness and other genetic diseases. The functioning gene that would be placed in Sam's eyes would not restore Sam's eyes, but would not halt further vision loss. But would halt further vision loss.
I look forward to the day when I could go in and have the surgery done. Dr. Stone. It's just something about him. He says he's going to get it done. Have the surgery done. Trust him. He's going to get it done. Dr. Stone. It's just something about him. He says he's going to get it done.
Press enter to enable the narrator.
RNA with another amino acid, and that amino acid binding to the existing chain, and on and on and on and on and on. Sometimes for thousands of amino acids to make a single polypeptide chain, for example. This whole word is basically just the names of the amino acids in the sequence and the order in which they occur in the protein, all 34,350 of them. But before we can make our own hot pockets, that string of amino acids becomes my muscle tissue. We have some folding to do. That's because proteins, in addition to being big, can also contort into very complex and downright lovely formations. One key to understanding how a protein works is to understand how it folds. And scientists have been working for decades on computer programs to try and figure out protein folding. Now, the actual sequence of amino acids in a polypeptide, what you see scrolling along down there, is called its primary structure. One amino acid covalently bonded to another, and that one to another, and to another in a single file. But some amino acids don't like to just hold hands with two others. They're a bit more promiscuous than that. The hydrogens on the main backbone of the amino acids like to sometimes form bonds on the side, hydrogen bonds, to the oxygens on amino acids a few doors down. When they do that, depending on the primary structure, they bend and fold and twist into a chain of spirals called a helix. We sometimes also find several kink strands laying parallel to one another called pleated sheets. All those hydrogen bonds and pleated sheets are what make silk strong, for instance. So in the end, our promiscuous amino acids lead to wrinkled sheets. These hydrogen bonds are what help give these polypeptides their secondary structure. But it doesn't end there. Remember the R groups that define each amino acid? Well, some of them are hydrophobic. And since the protein is in the cell, which is mostly water, all of those hydrophobic groups try to hide from the water by huddling together. And that can bend up the chain some more. Other R groups are hydrophilic, which if nothing else means that they like to form hydrogen bonds with other hydrophilic R groups. So we get more bonding and more bending, and our single file line has now taken on a massively complex three-dimensional shape. It also explains why I can fix my bed head by wetting my hair with water. The water helps break some of those hydrogen bonds in the keratin, which relaxes its structure. That way I can comb it out, and when it dries, those bonds reform, and voila, perfect hair. All of this shape caused by bonding between R groups gives our gives our polypeptide its tertiary structure. So now we have a massively contorted polypeptide chain, and it actually contorts very precisely. Sometimes just one chain is what makes up the whole enzyme or protein. In other proteins, like hemoglobin, several different chains come together to form a quaternary structure. So quick review of structure. The sequence is the primary structure, the backbone of hydrogen bonds within the sheets and spirals are the secondary structure. Our group bonds are tertiary, and the arrangement of multiple proteins together give the quaternary structure. These polypeptides are either structural proteins, like the thing at the bottom here, that you can find in your muscle or in my hot pocket. They might also be enzymes. Enzymes like do stuff. They can cut up biological molecules like I do with the chef's knife. They can mix stuff and they can put stuff together. So from that one recipe book, we get all the ingredients and all the tools necessary to make me, which is better than a hot pocket. Would you all agree? Now take your time with this stuff. Feel free to watch the episode a couple of times, because next week we're going to talk about how cells swap all of this genetic information through reproduction. Thank you for watching this episode. By now you should probably know how this works. You can click on any of the links over there and it'll take you back to that point in the show, as long as you're not watching on your cell phone. It doesn't work on cell phones. I apologize for that. Thank you to everyone who helped us put this show together, and thank you to you for watching it today. If you have any questions about this episode, please leave them in the comments below, or you can get us on Facebook or Twitter. And that's all. Goodbye.